Okay, I'm so excited right now to be here with my friend and colleague, Dr. Andrew Kaufman, who has been really leading this movement of awareness around how the dominant narrative around COVID being this lethal, invisible particle that sprung up in Wuhan, you may a wet market or some kind of bioweapons facility, taken over the planet, required the complete cessation of most social economic activity on the planet, as well as suspension of our civil rights and the implosion of the economy, the injection of a global governance structure that some would call the New World Order. All of this being done on the back of this invisible particle that we are all convinced was isolated, identified, and that is being tested everywhere. People are dying, in the droves from it, and yet from the beginning, this man that I have great respect for and has incredible courage, has been telling the truth from the perspective of the science itself. And there's just so much you've already done. I encourage everyone to go to your website. We'll say it again at the end. And also go to Questioning COVID, where we have a lot of your videos already set up for the red pill of going down the rabbit hole, asking, are germs real? What are viruses? Can they kill a person? And then, of course, the topic of exosomes, where, just to give people a preview, your discussion on how COVID-19, if you will, is actually an extra cellular vesicle called an exosome, is absolutely compelling and just blows my mind. So anyway, I'm so excited to have you here today, introduce my following and readers to your work. And, the sky's the limit here. I'd love to know what's top of your mind on this topic and what you'll be sharing today.
Well, thank you so much, Sayer. And, I can't believe we haven't done this sooner, because we've had some really excellent conversations. And, you're one of the few people that I can really talk about this, on a deep scientific level. And that's really helped me, develop and clarify some of my ideas about things. So I really appreciate this collegiality. And it's great to be here to talk about this. And, what I see going on is that over and over they're putting out more and more papers, basically using the same exact experimental methods and sort of calling it, more evidence. And so, periodically, because I put out this, sort of challenge, if you will, early on, I said, if anyone, has a paper that they think proves that this virus exists or that causes a disease, please send it to me, because I want to have a chance to look at it and see, because I'm willing to change my mind if new evidence does come. But what I've seen over and over again, and there are papers, where they claim to have sequenced the genome, and then there are papers like the one we're going to discuss in detail today, which claims to prove causality. And in this case, they again use Koch's postulates, which is a standard that I've talked a lot about. So as these papers keep coming out, it's great to, have a chance to look at them in depth. But what I'm really seeing is the same fraudulent claims, sometimes outright lies that I'll demonstrate, as well as, using the same kind of experimental techniques that don't adequately do what they set out to do.
Can I ask you then, are you then saying, for those who are new to this topic, that they may not have actually even sequenced the genome of this virus that has effectively shut down the world and reorganized the world, that for so many people, they just can't even consider that that's even possible, because it's a real thing. And of course, they've done this probably in labs throughout the world, and they haven't proven causality? So these are great things to bring to the fore, and that is actually exactly what you have been doing, is showing that the emperor wears no clothes. There is no evidence apparently to prove those things. Is that right?
Yeah, absolutely. And the genome issue is one. I don't want to get into it in detail today, but what I would accurately describe what they have done is that they've created a theoretical computer model of a genome of a theoretical virus.
Huh.
And I'm working on actually a white paper, like a scientific type paper, discussing that with Kevin Corbett. We're collaborating on that. So hopefully that will be out, in the next several weeks. I want to do a really good job. But, if anyone wants to look into next generation sequencing, that's the main technique that they're using. And that's a really powerful technique to sequence a genome of an organism which you already know their genome. So in other words, you can use it as a shortcut, and then you can compare it to the reference genome, right? And then you know you have that organism. So it's great if you like trying to look for a mutation that may be associated with a disease, because you already know what the sequence is, and then you finding where the sequence has changed. But to use it to show a brand new organism that's never been sequenced before, there's no way to tell what you've got, because you have no reference to compare it to. So that's why they use the computer modeling to essentially just create a genome out of, from scratch. And that's why different labs that have done this have genomes that are different lengths and different sequences. They say that it's a mutating virus, but really what it is they can't reproduce the experiment because they're just theoretically constructing a novel genome.
So out of curiosity then, when we hear about there being over 100 coronavirus types that have been identified, theoretically, those themselves could be really exosomes or cellular debris or nucleic acids that were misidentified as being viral, but then were synthesized into this picture that then, like you're saying, it wasn't just found there and sequenced, it was synthesized because it's, I guess it's such at a nanoscale anyway, it's all deduced on some level. I don't know, maybe you can.
Right, well, Sayer, they do these like two different parallel experiments, which they say is, proves that a virus exists or that they isolated it. And one experiment is this, what we're talking about with, they pull out a few genetic sequences that, sort of match something else in their database that's a virus. And they say based on that, it's a virus, right? But if it's a new virus, they don't really know because they have nothing else to compare it to. And it's not like they took the virus particle in a pure form and took the genome out and sequenced it. That would be a real experiment.
Wow.
But they just do these fragments. And then the other experiment they do in parallel, they call the isolation, but what they really do is take some body fluid from a sick person and put it in a cell culture with several toxic chemicals, and then they see particles. And most likely the particles they see are exosomes because they basically create a recipe in that cell culture to induce exosome production. And so that's probably the predominant particle, but it's really impossible to tell for sure because we didn't get all of those slides and be able to look at all the images and then try to purify out particles from there and characterize them. Then we could tell what they are.
Okay, so for people that don't know, when a cell undergoes death through injury or apoptosis, there are these apoptotic bodies that are like extracellular vesicles or hard to distinguish because there's so many of these different viral particle-like entities. Is that right?
Yeah, well, inside of our cells, there's a lot of different things. And especially if you have a cell that's poisoned, there would be poisonous things inside the cell. Mm-hmm. And if cells just break open and release their contents into an organism, it causes all sorts of problems, right? It damages other cells, causes an inflammatory response, etc.
Yeah.
So your body tries to prevent that. And when cells start to get damaged, they basically start to break down and their membranes pinch off into little vesicles. Wow. And that kind of contains the toxic chemicals inside so that it could be, either pass a message on or be eliminated from your body without causing more harm on the way out. So when you have this kind of toxic soup in a cell culture, you get all different kinds of particles. So there could be apoptotic bodies if there's a programmed cell death that becomes initiated. But there's also like all other kinds of little vesicles and particles, and they're very heterogeneous. And then there's also you have exosomes, and so at different points in the dying process, right? Initially there might be more exosomes, and then if the cell continues to die, it might be other more just necrotic type particles. But it's without doing a chemical analysis of the particles themselves, you don't know what they are. And that's what they do in all the exosome experiments. Like they purify the exosomes, then they do chemical tests to see what markers are on the membrane. Then they can extract the genetic material and sequence it, like end to end, like using micropore sequencing. But when they do that, then they can really characterize what that particle is. But in these experiments, they just show images and they pick out a particle that they say looks like that particular virus, but they don't actually characterize it or purify it, so you don't really know what it is. And I think most likely it would be exosomes because they share the most microscopic properties with what they call virus particles.
That's fascinating because I was listening to a Hopkins University virologist discuss the COVID-induced cytokine storm, and he himself discussed how exosomes are part of the release that occurs when cells are damaged, which then causes this inflammatory response that can cause a lot of damage. And yet, even the virologists themselves are saying, well, exosomes are actually implicated in what they're calling COVID-induced pathologies. And the second thing is if you have cells that are being damaged, releasing all of these bodies, and you're using a PCR test, right, that's amplifying nucleic acids, then theoretically it would be causing all types of false positives, right? Because
yeah. Well, Sayer, actually right out of this paper that we're going to discuss, they describe how they prepare the sample for those tests. And essentially what they do is they take the lung fluid, right, which has all kinds of sources of genetic material because it has various human cells, lots of bacterial cells, and perhaps, other virus genes that could be expressed in there. And they do something called homogenize it, right, which is they put it in this machine that physically agitates it. And what it does is it basically disrupts all the membranes of any cells that are in that soup from the lung fluid. And so it releases all the genetic material.
Wow.
Now, since they're testing RNA, every organism that's expressing any gene would have RNA of that gene and other regulatory RNAs and tRNAs and et cetera. So all of that would be in there. And when they're using the primers, and we know the primers have a lot of homology with human sequences. So there was the one, from the Pasteur Institute protocol that was identical to a chromosome 8 sequence. And then when I did some analysis on the protocol for the United States, I found a lot of primers had something like 18 out of 24 homology to human sequences. So, if you amplify the test enough, you could easily get false positives from those. So you can demonstrate these things, in this toxic soup, but you don't know where those genes came from. Wow. Because they didn't come from a single organism. When they sequenced the human genome, right, they had an isolated human. They didn't have, human cells mixed up with 100 other cells. Right, they got it directly from the human cells, and that's the standard I would expect, for a virus or any other organism.
Wow. Okay, so that definitely challenges the veracity of those tests, of course. And okay, so when I reached out to you before the interview, what we'd done in the past is we'd have these conversations, and I was thinking, wow, what a wealth. Every time you speak about this topic, I just want to broadcast it globally because it is that important. When people understand that this narrative that has been weaponized now, in fact, I like to discuss germ theory in terms of its political application as a weaponized instrument for control. When they realize that that's really all it is, it's not actually based on science and evidence and electron microscopes confirming the presence of these sequences or viruses, it's really, so thank you for, again, providing this insight, because only someone with your background, I think, can really navigate and translate for us in this way.
Well, I appreciate the opportunity, and it's just amazing because, I have done some research earlier in my career and designed experiments and learned the basic principles of research, like have a control group and things like that. And how to do my own statistics even. And in fact, I'm going to do a little bit of analysis in this article. And it's just astounding to see that these basic principles that were kind of drilled into me by some very good mentors, and I'll give a shout out to Bruce Way, especially, they're just not followed in these experiments. And yet, they're getting published, like this paper is in Nature, right? That is one of the toughest journals to get an article in. So it's just quite astounding. And, sometimes I can't believe it myself, but over and over again, I see the same patterns. So, thanks for letting me talk about it here.
Yeah. And the context is that, you've been helping me and others understand that the Koch postulates have never really been fulfilled in the least. And you keep going through these studies from Nature, high gravitas journals, and every time you go down the rabbit hole with us in your videos, it's clear that they haven't fulfilled any of them. And in this case, I sent you this paper that one of my colleagues was like, no, this is wrong. They're proving causality. COVID causes this. And I sent it to you and you did your magic. And here you are now to show us that no, actually, there isn't any evidence yet to prove that there's causality here. Is that right?
Well, Sayer, there's really not a lot of magic in it, but they've done a little trick in how they change the format of these articles. So always, I've read lots of scientific articles over the last 20 years, and they have this format, right? And it's like, abstract, introduction, methods, results, discussion, conclusion, every single time. And what they've done now is they took the method section out.
Wow.
And they put it in the back as an addendum.
Huh.
And it's like they don't want you to read that. And I'll tell you that is the most important section to read of any scientific article, because that tells you what the experiment actually is. And you can scrutinize that and sometimes say, well, this experiment is not even powered to determine this outcome.
Oh my God, that's why they call it the scientific method, right?
Exactly. And so, I go to that first and start reading that and then see, okay, what conclusions can be drawn from this experiment?
Oh, wow.
And then I look at what they said and, see if it matches up. And so many times it doesn't. But so I think a lot of people, when you read the conclusions or the abstract of this article, it sounds really impressive. But when you go to the method section and see what they actually did, you're like, whoa, wait a minute.
Yeah, right on. Yeah, do you want to show us?
So let me bring up this slideshow that I prepared here.
Oh, wonderful. You won't get this education at any Ivy League university. This is a very special opportunity.
All right, so hopefully you can see that okay. And so this is the paper we'll discuss, and I'll just show you the actual paper here for the audience.
Great.
The camera. Okay, so it's got a very scientific title, The Pathogenicity of SARS-CoV-2 in ACE2 Transgenic Mice. So I'll just break that down a little bit. Pathogenicity means basically causing illness. SARS-CoV-2 is the virus that they say they've identified and they say causes COVID-19. And then we have ACE2 transgenic mice. And so these are actually not normal natural mice, but they've been genetically altered to express the ACE2 receptor, which they say is the receptor that this alleged virus attaches to our bodies. But the thing is that mice don't normally express this in their lung tissue or other tissues. So these mice may not have normal physiology. They may be susceptible to illnesses and stuff. So really, that right there actually kind of disqualifies because of what Rivers said, and we'll get to that exactly. But nonetheless, I think we can still look at this study and learn a lot from it. So in the first sentence of both the abstract and the text of this article, it says outright that this virus causes COVID-19. In the abstract, it says severe acute respiratory syndrome, SARS-CoV-2 is the cause of coronavirus disease 2019. Now, that's a very bold claim, right? Because is caused. So they're stating it outright. It's like a foregone conclusion because the paper is actually setting out an experiment trying to determine that. But they give these two references here on the slide, 1 and 3, as basically the source of a conclusion, a scientific conclusion that there's a causal relationship. So I went and looked at those two references, and they were both familiar to me already. One, and this we're going to come back to this reference again later, is one of the first papers from the New England Journal of Medicine, and it's the Chinese study where they say they identified the coronavirus. But in their discussion section, what they say is that there's likely to have been the cause. So not is, but likely to have been, not even very likely, but just likely. And then they also say their study, they admit, does not fulfill Koch's postulates. And then it provides evidence implicating the virus, okay, just implicating. So you see that that's not even close to the same thing. And this, is a somewhat appropriate conclusion if they believe that they really isolated a virus. Now, if we go to reference 3, we see even weaker claims there, where they say that the virus is only associated with a disease. Okay, so we see basically right off the bat, this paper is lying to us outright by saying that it is caused by, as a declarative statement. And this fits a pattern because I've seen this in several other papers that in their introduction section, they make this claim and they put a reference, but the reference does not conclude that at all. So it's essentially a fraudulent, and then this rumor gets spread around and is used to design policy after, because they just accept that this is a cause, but there's no scientific paper other than this one that in its conclusion says that there's a causal relationship. But this one, we'll see if that conclusion stands up or not.
Yeah, and if I could just jump in and say that that's one of the ongoing, sort of psyops against vaccine-induced injury, that they say, oh, well, correlation doesn't equate to causality, but they give themselves a pass when it comes to these sorts of papers and their declarations.
Yeah, and in the psychiatry literature, you see this as well. You can see hundreds of papers where in the introduction it says that, since the modern era of psychiatric drugs, it's been like a great breakthrough. But actually, if you look at the disability from psychiatric illness, it's gone way up since those drugs came on the market. So, where does that claim come from? There's actually no evidence behind it at all. It's just a rumor. So this is the statement in the conclusion of this paper that we're going to go back to the experiments and see if it actually met this claim. But it says their results, together with another study, and we'll get back to that reference 1 later, completely satisfies Koch's postulates and confirms that it's a causal relationship, essentially. And for Koch's postulates, so this paper, different from other paper, it doesn't say that it uses Rivers' criteria, but it does reference the Rivers article. So since it says it's using Koch's postulates according to the Rivers article, which is reference 7, I thought we'd look at what Rivers said. So one of the first things Rivers said is, it's obvious that Koch's postulates have not been satisfied in viral diseases.
Wow.
Okay, right out of that paper. But what he did do is he said that his definition of Koch's postulates comes from these 2 quotes from Robert Koch. And the first one is from an article on tuberculosis, which essentially says it is not sufficient to establish only the concomitant occurrence of disease and parasite, but the parasite must be shown to be the real cause. Okay, so this causality is really important. And this can be done only by fully isolating the parasite from the body and all products of disease which might be considered as having a deleterious effect. So this is really important. You have to take the virus that causes the disease from a sick person, but have it be purified. So it can't contain other cells, other genetic material, or chemicals that have a deleterious effect on health, right? So that could include antibiotics, breakdown products of cells, other things. And then it has to be introduced into a normal host. So a normal host, right? Not a transgenic host, but we'll let that one slide for now. And then the second quote Rivers uses is from a speech he delivered in Berlin, and says, first, the parasite occurs in every case of the disease in question. So that's really important. And then secondly, it occurs in no other disease. And then once again, to reiterate, after being fully isolated from the body and repeatedly grown in a pure culture, can induce the disease anew. So in other words, create the same disease in whoever you put it in.
If I could just ask you, so basically the first postulate is that you should be able to find this singular pathogen in that individual without it also being found in healthy individuals, which sort of, they didn't know about the virome at that point in time, or the fact that the coronavirus family, if you will, of exosomes or viral-like particles are actually naturally present in most humans. So is that true? And that's implied here too, yes?
Yes, and I think this more easily applies to a common situation like strep throat. Right, so if we do throat cultures on people without a sore throat and they have streptococcus and they also have it in a sore throat, then it can't be the cause of the sore throat, right? According to Koch. But yeah, you're right. It's hard to be as stringent on that, but nonetheless, this is what the article claims to be using. The other thing is grown in a pure culture. That's impossible for viruses the way they say they work because they depend on a host to replicate, right? They have to invade a cell in order to reproduce. They can't reproduce on their own. So unlike bacteria, which can be grown in a pure culture, but there's an adaptation for that. So I've just codified from those two quotes because this is really the test that the article that we're discussing claims to use. So first, the virus is present in every case of the disease. Notice I didn't put that it's not present in healthy people. The parasite should be fully isolated from the body and all products of disease, i.e. purified. That's super important. 3, the pure culture, I'll say it's not possible for viruses. And then 4, the isolated parasite produces the same disease with all of its characteristics in a normal host. That's really important, all of its characteristics.
Can I just ask you really quick? So far, to your knowledge, this has not yet been done for what they're calling COVID-19?
Correct.
Okay, great.
And this is the main article that claims that it has been done. There is one other article using monkeys, but also I've done an analysis on that, and it's very similar to this one.
Do you have a name of the video on your channel that you do that analysis with for the viewers?
Oh, that one I just did for myself.
You're really passionate scientist. I love it.
I mean, every article that comes out, I look at. So, wow. But I don't, make a video on everyone. I'd be doing that all day long.
Yes.
So here is how rivers accounted for the inability to grow them in a pure culture. He said, and he also said it's important to show that it's actually the cause. He reiterated that even with his modifications. And he said basically that you have to can get transmissible infection in susceptible experimental hosts by means of inoculation of material. So that's basically what you take out of the sick person is the material. Free from ordinary microbes or rickettsia, which are parasite, obtained from patients with a natural disease. Okay, that's a critical thing, from the natural disease. In other words, right from the patient's body fluid. Okay, so let's look at what they did in this study. So what they did is they took these transgenic mice and they had 2 what they would call control groups. One group was regular, what they call wild type mice, so they didn't have the inserted ACE2 gene. They also, gave them whatever they say is the virus. And then the third group they called mock infected. So they were, I think they were also transgenic mice, but they were not given this concoction. They were instead just given phosphate buffered solution, which is just basically like water that they use in the laboratory. So it wasn't a true control because they didn't give them something from a body fluid from a healthy person, right? They just gave them basically like saline. So it's a sort of control. And then what they did is they gave them what they call the virus, and I'll tell you what that is in a minute. So here, just to point out again, together with previous clinical studies, number 1, so we're going to talk back about that Jew article again. So the first postulate that the virus has to be present in all cases. Okay, so that means there actually has to be a virus. So it uses the Jew study to prove this point, but in that Jew study, there was no virus particle directly from a patient that was demonstrated or identified at all. Okay, the only particles they showed were from a cell culture, I think using monkey kidney cells in this case. So the second point is that as we talked about before, they had these short sequences of RNA, but they didn't know the origin of those because they were from lung fluid sample with lots of RNA. So that's not conclusive. They showed the microscopic images of the cell cultures like we talked about before, but we don't know what the particles they are that were shown because they didn't purify them and characterize them chemically. And then this study only had 3 patients. So that's not really enough to say that it's everyone with a disease if you're only talking about 3 people. So this doesn't satisfy Koch's first postulate at all. So now we get to what the material is. So this is, the second postulate is that we use this purified material free from anything else that could be harmful. Now, what they did to get this material is, so they took that lung fluid from the patients in China, from 3 patients. They mixed that with monkey kidney cells and grew it in a culture until there was basically those cells were dying. So at that point, and then they added several things to it. They added bovine calf serum, which is basically part of the blood from baby cows, so a different species, so you could have all kinds of reactions to that. And then they added some antibiotics, which are shown to make exosomes and also be toxic to cells. Mm-hmm. So they had this mix of dying monkey kidney cells and all the particles associated with it, and these other chemicals, and the lung fluid grown together. And they basically took that, and that's what they inoculated these experimental mice with. Hmm. Okay, so it's not directly from the patient with disease, it's grown out of the cell culture. So it doesn't really, so it's not purified, right? And it's not from a patient with natural disease. So it doesn't really satisfy Koch's second postulate at all.
Yeah, and those out there who want to better understand, the connections, is that if you have a cell and you're harming it, it's basically compromising its resilience. The tensile strength of certain healthy cell membranes is equivalent to steel. So to think that a viral particle that isn't technically alive is somehow going to tackle it and penetrate it if it's healthy is absurd. And when a cell is compromised and harmed, then it undergoes this process of communicating perhaps horizontally to other cells through exosomes. Hey, there's trouble here, activate your detoxification pathways. Or it might start breaking down, releasing apoptotic bodies and other extracellular vesicles, which will be misidentified as viruses. So anyways, that's a context for...
Sayer, even more simply, you could just think about it as rotting flesh.
Yeah.
Right? Because that's essentially, it's from a sick person, it's their excretions, so it basically contains dead and dying cells and material, right? And,
yes.
You wouldn't want to go near it. It'd probably smell bad. It'd be discolored.
And that's right? That's the beautiful forest floor metaphor anyway, is that the reason why we see, quote, infection or opportunistic overgrowth of the virome and the, bacteria, fungi, is that it's helping to serve the degeneration of the tissue so that it can be regenerated through stem cells, et cetera. So.
Right. And by the way, if you wanted to do this step properly, it wouldn't actually be that difficult. And it's been done with exosomes. It's been done with viruses and bacterial cultures and algae and such. And what you do is just take the lung fluid and you run it through a filter first, which separates all the cells out, because these particles that they're looking for are way, way smaller than any cell. So immediately you get rid of all the cells, any bacteria or human cells. And then you do what's called a density gradient centrifuge, which is a pretty standard thing, one of those machines that spins it around. And all the particles of the same density will be in a tight little band in the test tube and separated from all the chemicals, because the chemicals will be much less dense. So they'll be far away. And then you can just use a syringe or a pipette to take out that band of those particles. And then you could give that to the mice, right? And then that would be, and you could look at it under the microscope and demonstrate that it was a pure homogeneous mixture of particles.
Wow.
Right? It's well, like if you just get a standard microbiology lab textbook, right? It'll give you this procedure. I mean, it's been done, many, many times. There are hundreds of bacteriophages that have been purified this way.
Wow.
Right. So it's tried and true, but they didn't do it.
Yeah. And those who are, aware of the dangers of the vaccine schedule and the origin of vaccines, is they're basically taking filthy tissue, and they're injecting that into healthy individuals, creating a quote immune response. And that is the basis for at least the first iteration of vaccines that relied on live or attenuated, basically filthy tissue.
Well, Sayer, it's like the procedure they use to create this is almost exactly the same as how they manufacture vaccines. They put this material, maybe not directly from a patient, maybe it's already been cultured, and then they put that in another culture. And when they say it's been attenuated they basically just reculture the material so it dilutes the toxic aspects of it each time.
Amazing. That seems to be the basic mechanism here is you get people sick through all types of means, it's obvious, and then you blame it on invisible particles like COVID or any other particle they want to come up with.
Well, that's the weaponized strategy that you talked about with germ theory.
Yeah.
So we can just get to the fourth postulate, because I think this is also really, really telling. And so this is basically the results. Did it produce the same disease with the same characteristics in the experimental host? So, we've already shown that they didn't actually get a purified virus. So we could just dismiss this part, but I think it's really interesting to look at because this is what they used to draw their conclusions. And so we see the three different groups of mice, and they said clinical manifestations, which is their way of saying symptoms, were recorded from 13 of the mice, and that's less than half, by the way. So even though they gave this to the mice, most of the mice didn't manifest any signs of illness at all. So they basically looked healthy clinically. And if we look at the numbers, so in the experimental group, so they're the ones that are transgenic and given this toxic inoculation, 37% had symptoms, and in the other two groups, only 20%. So I was like thinking, are these actually different? Because it's not a big sample size. There's only less than 20 people in each group. So I did a chi-squared test. And this is a simple statistical test of proportion that can tell you, is there a difference between those three groups based on the numbers? Like, is it statistically significant that those groups had a different result? And what it showed is that actually there's no difference. So whether it's 20% or 30% had symptoms, it's essentially the same. It could be 30% in a different group and 20% in that group, and the groups are no different in terms of expressing clinical symptoms.
Wow.
So in other words, the experimental group and the control group are not different from each other.
Wow.
So right there, you can throw that out, but there's more, Sayer.
Yes.
So if we look at what the symptoms were that they said were in the experimental group, they were weight loss and slightly bristled fur. Now, tell me, are those symptoms of COVID-19? I didn't see any articles that say bristled fur is a symptom of COVID-19, and weight loss is not listed.
Wow.
So COVID-19 causes respiratory symptoms primarily, right? Cough, difficulty breathing, and of course, fever, right? Those are the ones that have always been consistent. We've heard other things like it affects taste and smell, can cause nausea, et cetera, et cetera. But here we have no respiratory symptoms at all. We have weight loss. Now, weight loss, is usually a symptom you think of in things like cancer, right? Not an acute infection. There's not enough time to lose weight, right? But here, that's the primary symptom. So it's not the same characteristic as the disease. So we fail again.
It's amazing because this doesn't appear to be anything even resembling science, right? It really, obviously propaganda seems like an appropriate label. It's really embarrassing, isn't it, for the publishing industry and science in general for this to have been published and then, declared to be proof of something related to COVID's pathogenicity.
It is extremely embarrassing because it really just ruins any perception of integrity among the scientists in virology.
Wow.
And, perhaps it has broader implications. But, don't forget that Ioannidis put out the article, and I think it was in 2010, in PLOS 1, that, more than half of all scientific conclusions are false.
Yeah, I remember I reported on that. Yeah, at the time, it was really saying evidence-based medicine is nothing more than a coin's flip of certainty on pretty much anything. And the implications are, obviously related to some people call it the post-truth era we're in, and science is sort of leading the charge in that direction. It's just pure, fiction on some level at this point.
Yes. Yeah, it's amazing. And I think, in the virology literature, it's far less than half are true. Far more than half are false. as I'm seeing here.
Well, this is
So,
amazing. Yeah, please.
so we could just take this to one more level and look at the autopsy findings, because in this paper they did do autopsies on the mice. They didn't report the microscopic findings in the two control groups, only the gross pathology. And they said there were no gross lesions in the lungs, but they did look at the lungs of the experimental mice. So there are two, one of the big charades about this pandemic is that they didn't do autopsies. All the public health law says that if you have a public health crisis or a pandemic, you autopsy everybody to find out what's going on. But in this case, all the state health departments issued these memoranda or letters saying not to send anybody's for autopsy. So we don't have a ton of autopsy data, which we should have thousands and thousands of autopsies. But we just have a couple of studies in the literature. So, but these two studies are the main ones. One is from Italy, looking at 38 cadavers. And these are the main findings. And so I'll translate these into English for you. So diffuse alveolar damage, essentially alveoli are the little sacs where we exchange oxygen and CO2, and they're just basically damaged. So that would affect breathing, right? Mm-hmm. It's interesting that we didn't see any breathing problems in the mice, but in these patients, they all had respiratory failure or serious respiratory symptoms. Necrosis of pneumocytes, that means basically lung cells that are dying, right? And they probably look like they're fragmenting into particles, just like we described earlier. Hyaline membranes are something you can see under the microscope that forms in certain types of infections and conditions. Interstitial or edema basically is just fluid in the tissues, like swelling. Metaplasia, that's an interesting finding. That means an abnormal growth, like you might see in a cancer or a precancerous lesion. So it's a pretty specific thing. And then blood clots. And that was the biggest finding, right? Because we've heard in all these autopsy studies, they found basically blood clots in the lungs, and has led people to say that the disease should be treated with blood thinners, and not so much ventilators. So then we have the German study, smaller, but essentially the same findings. So the diffuse alveolar damage, hyaline membranes, metaplasia, and blood clots. So those four things are pretty consistent with both these studies in different countries, different data sets, different pathologists.
Is that the only 40 autopsies performed that you know of, or are there more?
There are some other ones, but I didn't easily find another scientific publication reviewing these. I know there's a small set from Louisiana that I saw in a regular news article, but I don't think it was written up for publication. But essentially, it also showed blood clots.
And if I could just zoom out for those who aren't already aware, the ICD codes were updated, emergency use codes, by the World Health Organization in March, where they basically removed any criteria for proving that a virus is present in the case of someone being coded dead by COVID. All they needed to do was have suspicion that it was involved, and they made it clear that if that there would not be follow-up autopsies, after the emergency diffused. So it's very suspicious. And if we have truly hundreds of thousands of people that have died, why are there not more autopsy studies? Yeah.
Well, because they would find other causes of death, most likely.
Exactly.
But these, all the cadavers included in these studies, were all had the typical respiratory type of illness, and had positive tests. Mm-hmm. So, at least they used some more rigor than the CDC guidelines for death certificate reporting.
And just out of real quick question is, obviously, like in Asia, there was already a lot of the use of masks due to pollution, which I think it's something like 5,000 people a day die as a result of inhaling pollution. And of course, cleaning solvents and disinfectants, it's like the equivalent of smoking a pack of cigarettes a day for those exposed as cleaners. So can any of these symptoms be caused by exposure to chemicals? Is that possible?
Yeah. Well, when you have pneumonia, essentially, can be caused by a variety of things, and definitely chemicals. It can cause by aspiration, like if you have throw up, basically, and then inhale some of the contents into your lungs, right? That can cause pneumonia. So there are many, many things that can cause this type of reaction. Absolutely.
Great.
I mean, I would say that it's never caused by microorganisms, but we can leave that for another discussion. But certainly these are common findings with pneumonia and pulmonary embolism. By the way, usually pulmonary embolism in regular pathology would be associated with bacterial pneumonia, not viral.
Okay. Mm-hmm.
So let's look at the autopsy findings from the study. So these are back to the mice that were given this toxic inoculation, and by the way, they put it in their nose. So they did one of those swabs way in the back of their nose, and that's how they, got them to be sick, but they didn't express many symptoms. So what it found was interstitial pneumonia with predominant macrophage. That's a type of white blood cell or immune cell infiltration. And so this is a bit different. The diffuse alveolar damage that I mentioned before can be seen in interstitial pneumonia, but in the human studies, they did not see macrophages. They saw lymphocytes, a different kind of white blood cell. But this was so important in the mice study that they mentioned it in the abstract. And it was distinct from the other, but not the most distinct, because there were no hyaline membranes found. There was no edema. There was no metaplasia, and there were no blood clots. So essentially, these pathologic autopsy findings were not showing the same characteristics as the human COVID-19 autopsy studies.
Got it. And that's postulate 4, so it doesn't fulfill that.
Right. So we have, to summarize postulate 4, first, we have no difference in symptoms between the groups, and the symptoms then secondly are totally different from the symptoms that they say are COVID-19. Right. Then thirdly, the autopsy findings are different. Yes. So at every level, we didn't produce the same disease with the same characteristics.
Wow. This study appears to be an example of science by proclamation, or eminence-based medicine, where the cult of authority, provides them this opportunity to declare that they found something that absolutely was not found. And then everyone believes them, right?
Yeah, well, because it's really easy to just read the abstract. It sounds like it's a solid experiment. It sounds well thought out, well researched, and it makes bold conclusions. So, it would be easy and very time efficient to just walk away from that and take it at face value. And that's what, probably 999 out of 1,000 people who would read an article like this do. Because, like you're a scientist or a doctor and you're following a certain topic, what happens is, you get like all these emails with all these abstracts of articles, and you figure out which one you're going to like read in depth. And it may only be 1 out of 10 or 20. And so mostly you're just skimming the titles and the abstracts, but you still take away, like if they make such a bold conclusion like this, why would you question it? And it's in Nature, right? I mean, it's pretty impressive on the face.
Yes.
But here are the conclusions that I drew from the paper. One is the first sentence of the paper was an outright lie. Mm-hmm. Secondly, not one of Koch's postulates have been satisfied, despite claiming that they've all been satisfied. And, despite taking a toxic liquid from a culture of monkey cells grown with infected lung pus and poisonous chemicals and putting it into a bunch of mice noses, less than half of the mice showed any symptoms at all. And no mice developed any symptoms or autopsy findings that were similar to COVID-19.
Wow.
I wanted to spell that out so you could get a graphic picture of what they did. These mice were pretty resilient.
As far as I'm concerned, you're like the Braveheart of science at this point. You just pretty much took a shillelagh and knocked down a whole army of trolls, which is basically what's happening right now through the media colluders with this, agenda, this psychological operation which they're running. This is amazing. I mean, this video, if you're watching, should be shared far and wide with anybody who has consumed the Kool-Aid, it's amazing.
Yeah, it's just it is so amazing to see that that you could criticize this to such a degree that it's basically completely invalid. It's never what I expected as a child growing up and wanting to be a scientific researcher or a doctor.
Well, that's the beauty of this, is that you are a scientist. You're both showing, the darkness here, the absurdity of what they're saying is science, and you're showing us that you can apply a method, and there is logic, and there should be evidence, and there is hope for science, again, returning to being a method of discovery and validation. But what it's become, unfortunately, as you've seen, is really a political weapon. And so I'm just hoping you might have a few minutes to zoom out and provide, after the many months of your, just dedication on this topic, some perspective on what you think is happening and where we're going, and what does the future hold? Because so many of us are still under the thumb of what are fundamentally draconian regulations and economic assault or implosion. And anything you can provide us would be so valuable.
Yeah, sure. Well, what we're seeing here is basically a healthcare-based technocratic future developing, where, all of these policies that essentially amount to having government or, whatever is in charge, whatever entity in the future is going to control all aspects of our lives. And they're going to use this threat of disease and of infection as the main mechanism to get our cooperation. And we've seen this happen already, right? Because they had some very scary predictions at first and said, for 2 weeks, we're going to just stay at home and be really careful. And now it's been more than 6 months. And we've seen, right, that the lack of death around us, and the lack of disease around us. And yet people are still scared enough and believe enough to follow, wearing a mask and social distancing and not having places open to full capacity, right? And we went a whole summer with no July 4th festivities, no summer festivals. Where I live in New York, we have the State Fair. It's the biggest thing in Syracuse every year, and it was canceled. So, and people are still kind of going along with this without, there are some people, like I do, where I don't follow any of these and I act as if things are normal, right? But there are very few of us out there. And the reason is because if they have this, I call it a dogma about viruses and germs, that they can basically appear out of nowhere, they're invisible, and they can, kill us if we're not careful. It can be used over and over and over again to control us, by the fear that it creates. And the simple truth that I've learned after doing this kind of research is that it's a farce, that there's really no scientific evidence behind it. And they have these kind of trick type experiments that they could do over and over again. Like if I had a laboratory and enough funding and the right equipment, I could take a few sick people who had an illness that was similar, and I could create a virus out of it, by basically just using the right primers and finding some sequences similar to a known virus. And then I could use the next generation sequencing. I could put it in a cell culture and show toxic particles, right? And essentially create that over and over and over again, anytime that I needed to use it for another purpose, right? This kind of problem, reaction, solution strategy. The problem is a new scary disease. The reaction is fear, and the solution is a policy to more tightly control, regulate, spy on us, right? Surveil us, et cetera, like we see with contact tracing coming out. And this kind of tyranny model or weaponized germ theory really has been around for over 100 years. And it was initially exploited by the Rockefeller medicine system as they took over medical education in the country in order to basically sell petroleum-based pharmaceuticals. And the only way to treat an infection, right, which is a germ invading us that we have no control over, is to go to a white-coated doctor to get a magical chemical that will cure us.
Yes.
And it's been used to control us in that way, right? And even though we know, like, for example, that people would have a cold or something they say is caused by a virus, they would demand antibiotics, which are for bacteria, from the doctors. Mm-hmm. And everybody, I think these days, every single person already knows, right, that you can't use an antibiotic for a cold. Yet, how many people still go to their doctor or call them up? And, that doctor will say, if I don't issue the prescription, they'll just go to somebody else who will, and they'll lose all their patients. So they all do it, right? And it can't possibly work, but we still do it and we pay for it, the privilege of doing it. And so I think the most important thing that we need to do is to show that this paradigm is really not based on any science. It's, completely vacuous. And we need to abandon it because it is being used to manipulate us in tyrannical ways. And we can see that there are ways already in existence. Many of them have been around since antiquity to allow our bodies to heal naturally. And it's way more successful than what you see in the allopathic health system. And I know, you have direct experience with that as well. So I want to bring about this, if in my small contribution, this like paradigm shift to a new type of way of looking at health that can actually harness our body's natural ability to heal, which, boy, it has from almost anything. We just need to support it and in the right way and not get in its way. So that's really kind of how I see the big picture of things. And by exposing this kind of fraudulent science, it helps to crumble that, false paradigm of germ theory.
You're so inspiring because, you could have the background of being a scientist and helping us with understanding the science not being correct. And you can see the vision of, a very dark future, descending. And yet you also are affirming what I know many of our followers are really already aware of, which is that our body is really a profound miracle. And of course, you're featured in the Thank You Body Rally video, which we're so honored that you are participating by sharing just the opportunity we have to, put forth our stories and to show up with full sovereignty and basically saying no to the agenda, just not wearing a mask, okay? If that's what you feel is going to be the right thing for your health, then that's absolute in my opinion, So anyway, I so appreciate your work. And, if there's anything else you can share just to wrap this up, first of all, how can people find you and follow your work, and what are you up to? And anything else you want to leave us with?
Yeah, sure. I appreciate that. Well, certainly, check out my YouTube channel and my website. I'm sure we'll have the links below.
Yes.
But I do have a couple of exciting things coming up I wanted to mention. One is on October 10th and 11th, there's going to be a live in-person conference called the Red Pill Expo, which is put on by G. Edward Griffin. And there's a great panel of speakers, like not just me, but Carrie Rivera is going to be there, and Del Bigtree, and many others. So it's like really exciting to actually have an in-person opportunity, mask-free, to get together and talk about this stuff. So I encourage people to check out the redpillexpo.com. And if you're at all able to come, and it's in Jekyll Island, which is where the secret meeting to plan the Federal Reserve was, so it has special significance. Please consider coming to that. And then I am going to be at the rally that you talked about with you in person, because my big project that I'm working on is a documentary film where I'm basically trying to show that germ theory is really a hollow theory, but also introduce terrain theory, which I feel is more accurate, and people already know about it because we know about the microbiome. And terrain theory is just the natural extension of what we already know and accept about our body's microbiome. And using healing modalities based on terrain theory, what you see is people healing from all sorts of diseases that they tell us are incurable. So I'm really excited about that project, and I'm collaborating with Marcy Cravat, who has done some beautiful documentaries in the past. And we're going to be coming down to Miami to interview Dr. Brogan and yourself. So I'm really excited about that project. It's not going to be completed for a while, but I'm spending a lot of effort on that. So wanted to mention it. And then lastly, this week I'm going to be, for the first time, introducing a health product that I'm going to be selling, and it's Shilajit, which is a trace mineral supplement based on basically the decayed minerals from an ancient forest. And they crop up through the ground in the mountains. And I've found a great source in Utah. And it's extremely high quality Shilajit, and it's hard to find that from Russia and India because there's lots of corruption in the businesses there. So I'm really excited about that, and I'll be putting out some more information this week about it.
Wow, that's super exciting, because I've been sort of keeping aware of Shilajit, and I know there's been some issues with potential heavy metal contamination. And
yes.
Yeah, I did not realize there was a source in North America.
Yeah, well, this is pretty not well known.
Wow. Yeah.
But it's like there's a single guy who harvests it and does the initial purification, and I get it from the person who does the rest of the purification process directly, and it's really, really high quality. And one of the things that happens is that sometimes where it's harvested, there are other mining operations, and they have runoff, and in those countries like in Russia and India, they might try to mix it with water right on the mountain, and they get the contaminated water, and that adds heavy metals to it. So that could be, one of the problems. But this has well below, like orders of magnitude below safe EPA levels for all the heavy metals. So it's really, really clean.
I've got really close friends, colleagues who are big fans of it, and I've just stopped even thinking about it because of the metal issue. So I trust you and your source, so I'll definitely want to personally try it out. And
also, the certificate of analysis is going to be on the website as well. So, because I want to be very transparent and let everybody see, this is exactly what's in here, so that there's no questions about quality or toxicity.
Beautiful. So first of all, I am so excited about this germ theory documentary. I mean, I just can't tell you and those who are listening how important this is. So get ready, because this paradigm is shifting, and there is a beautiful new world waiting on the other side, thanks to your work. And, thank you so much for your dedication for doing this. And everyone, follow Dr. Kaufman, get on his list, and participate in these events. And of course, you'll be joining Kelly and I here in Miami. October 16th is the rally. They're popping up all around the world at this point. We're only day 2 of launch, so we're super excited to have you involved as a friend, a brother, a colleague. So thank you so much for your dedication.
Yeah, I can't wait.
Awesome.